I think this is the first time I've ever chaired a panel in which the speakers have finished early. So congratulations to them. I have a notice that the chairs were given to read before the session, the questions start. So let me read that to you now. An important notice about IOM National Academy workshops, in contrast to consensus development conferences. This will be a standalone workshop and is not part of an ongoing study by the IOM. A brief overview summary of the presentations and discussions made during the workshop will be prepared by a rapporteur, who's here, and will reflect what transpired at the workshop. Very important to recognize that for the discussion we're about to have. It will be prepared in accordance with institutional policy and made broadly available to the public. All views presented in the report will be those of workshop participants, hence not the IOM. And the report will not contain findings or recommendations by the planning committee, and the summary will be made broadly available to the community. So I take that to mean that the next 15 or 20 minutes are extraordinarily important in terms of getting into the record what people think or what people want or what people would like to have heard. Nothing you think about but don't say will appear in the record. Therefore, it's critical that you say it now or forever hold your wish, as Derek and I would say. So as you think about the questions you'd like to put to the panel or the panel will put to themselves, it seems to me, to pick up on one of the comments made by one of the speakers, we used to live in a data-poor, opinion-rich environment, and now we live in a data-rich, opinion-rich environment. And how do we bridge that going forward to get to real implementable and important findings? And I think the other commonality I heard from the speakers, particularly from Jim, was that there are real difficulties in moving organizations to new approaches. And that raises the question, which Jim himself may want to answer. What do we need to know, and what do we need to do to convince people to adopt new approaches? So I'll open it to the panel first to discuss the questions that have occurred to them, and then I'd encourage each of you to raise your hand, and there are microphones, I guess, somewhere, other, or the outside that, if you indicate, will be brought to you for your questions. Panel first, Jim.
Well, given the introduction, I will now say that everything I say is my opinion only and not the opinion of Eli Lilly and Company. Getting people to do things as a good friend of mine who's a decision scientist says is always a matter of incentives. So no one likes to do things if all you take on is more risk. People tend to take on things when they see opportunity. And I think that's one of the things that's really bedeviling us when it comes to safety. We're getting better and better and better at what risk assessors call hazard identification. And we're getting worse and worse at real risk assessment. So as we move into a big data era, we get better and better at hazard identification. But there doesn't seem to be an incentive, and perhaps that's part of the clinical trial problem, there doesn't seem to be an incentive to do things differently because people see opportunity.
That seems to be sort of just what I said, an opinion-rich, a data-rich, opinion-rich problem. How do we bridge that? Because as we get more and more data, and it becomes more and more facile to analyze that data, we're going to continue to throw out more and more signals of one sort or another.
I don't have an easy answer for you, but let me give you one thing I think we have to do that I currently think we don't do and I find unacceptable. And that is, we really don't measure what we already do. We don't know where we're starting, but we have this perception that when a shiny new approach comes along that looks better than what we already do, that if we do it, what we don't currently know will all of a sudden become clear. It rarely, if ever, and I will say in my experience, never does. So we really have to, if we're going to change, in my case, if society decided that testing drugs in animals is no longer acceptable, that could be a decision society could make. And we're going to replace it with something. It is incumbent upon us as scientists to understand how well we're doing now, measure that, and then measure how well we're doing with the new technology and turn back to society and say, we're doing as well, we're doing better, or we're doing worse. These are the risks that we've chosen to take on, or these are the opportunities that we've gained by making this change. But we don't know where we are. We have a lot of anecdotal information, and that bothers me as a scientist, and it frightens me as we talk about collecting additional data. I think we need to get together and really understand what's our current benchmark.
I don't want to hog this, but do we have a mechanism, do you think, right now to share that data effectively? I mean, what you're saying presupposes that Lilly and academia and Merck and every other company are going to be totally open about the performance of their favorite testing right now. And I'm not questioning that, but I sort of really am. And I haven't even included the FDA or the people with the rich opinions but no data in that environment. So isn't that a massive step we have to take first?
Actually, I think doing it is pretty straightforward.
Yeah, but
the question is, what are the incentives
right.
to share the information? Now, again, not speaking for my company, I actually believe there's no competitive advantage in believing that your company has a better non-clinical safety assessment approach. Frankly, I don't believe anybody's doing it remarkably better than anyone else. And I also believe that the FDA will probably regulate to the lowest common denominator of behavior. So there should be an enormous incentive for everyone to share the data and improve. It's just very difficult to have that conversation.
Right, but every time I've been present at that conversation, the general counsel pops up with sort of suicidal commitments almost.
I've been in those conversations as well.
Okay, other comments from the panel first. Russ?
Yeah, I really enjoyed the presentations, and one of the things that it comes up constantly, you will notice I did not use the phrase big data in my talk, and I've struck it from my vocabulary because I think it does more damage than utility. But there are large data sets available, and they have the problems of causality and lack of causality. I am optimistic, however, that there's another use of big data, which is carefully culling it to find the embedded randomized trial that's latently present in the big data. So what I mean by that is, imagine a big electronic medical record with lots of data all over the place, all diagnoses, all kinds of tests, all kinds of missing data. But you have a question. You may be able to take that very large data set of electronic medical records, and you may be able to find a very small, beautifully curated subset of that data that basically simulates, in retrospect, a randomized clinical trial, because you say, I want to get cases, I want them to have these characteristics, and then I'm going to look for extremely well-matched controls. And the reason you'll find those controls, it's a big data set. And so in the end, you may have millions of patients, and you may wind up with 100 of cases and 100 of controls, and you may actually be able to learn something. And we've done this once or twice with some success. And so I just want to, in the name of putting hope out there, because these are hard problems, I think that some of these large data sets that look to be filled with noise, filled with bias, may be able to yield curated subsets of data that can be used for actual discovery.
Now, would you view that as hypothesis generating, or would you view that as definitive?
Right. So there's a good question. I think that if you are doing this on a large scale with no question, it's definitely hypothesis generating. If you have done something else in your life, in your scientific, intellectual, scholarly life, to come up with a hypothesis that X might be true, then I think you can make a credible case that going into the database, not checking K hypotheses, but checking one with this kind of approach that I just outlined, could be arguably probative, if not nearly probative, or, it's not 0 or 1. So what I think it would certainly do is move the bar in terms of your uncertainty greatly. So
would we need a system that forced you to declare the hypothesis before you
Yeah,
started?
yeah,
the
I think that
trial?
if you want to use it not for hypothesis generation, then you really have to have discipline because it is so tempting. You know, when you write code to do something, you can often run that code millions of times overnight for free.
Sure.
And we do impress on our students that that's treacherous because you're now doing multiple hypothesis testing and you're going to find more false things than true things. And so that's a discipline that's hard to automate because it's just so easy to press the button and see what happens. But it's very dangerous and it's bad science. And I think it's related to why we've had this proliferation of findings that are really not reproducible.
Derek?
Yeah, so my talk was made a little bit more incendiary. So yes, big data is excruciatingly cutesy. JAMA's trying to work out whether the B and the D should be capitalized or not, and they're currently deciding not to
Critical
capitalize
question.
it because they don't want to immortalize something that's sort of excruciating. And I think that this notion. There are instances where randomization might not be exquisitely helpful. So for example, even in a trial, in a way, for the very next patient, it's still a guess. And the randomization isn't necessarily outperforming some, again, big data techniques to try to create these sort of locally stable predictions. The issue is that it might not provide a broad class piece of information. And although I've spent years doing observational research, the only reason I was sort of pushing back is I don't like to think that we want to spend ages combing EHRs because we've already decided that it's too hard to randomize. That given the sweetness of the quality of information that can arise from randomization, I would rather that people inside healthcare systems using products around which they have intolerable uncertainty embrace the uncertainty and look for methods that lowered the bar on randomization, which is one of the reasons why things like response adaptive randomization takes away some of the risk for the patient. It's not giving them full control, but giving them full control to make a decision under which there's imperfect information isn't so much better than surrendering to mother being in charge of the green button, who's going to at least roll the dice for you proportional to the odds of success with the options available.
So you've used the term, many of you have used the term uncertainty quite freely. How easy is it to parameterize uncertainty? And is it just a gut feeling with big data?
I mean, it depends on the data set, but you often know more than you think you know. And once you do some of these exploratory kinds of exercises, you can get a feeling for what the magnitude of the uncertainty is. It may not be precise, but you can definitely have a sense of the magnitude, and that gets you started in the right direction, I think.
And then can we easily probe how uncertainty propagates through the model? So we've sort of spanned from preclinical safety all the way through to adaptive clinical trials. Are we just sort of segmenting, we do preclinical to get by the FDA, and then we use clinical trials to get a better answer for the patient? Or is there a way of using the former to inform the latter?
So in answer to your question, uncertainty really depends on what your default position is. If your default position is, in the pharmaceutical industry, good drugs are rare events, you're uncertain that stopping is a good idea because you might be throwing out a good drug. If you're a safety assessment scientist, now you're saying, well, my uncertainty is I'm not certain if we go forward, this is a good idea. Those are two completely different default positions. One is, absent evidence, I can't go forward, I choose to go forward. The other is, absent evidence, I have a good case that I should go forward, I choose not to. And I don't think we even resolve what our default position is when we try to deal with uncertainty. Now, I will tell you that the pharmaceutical industry behavior, and again, this is not the opinion of my company, is really well trained in the blockbuster model, the rare event. I don't want to miss the rare opportunity that pays for all the mistakes I made in ignoring my probabilities. So Those are two completely obverse views of how you deal with uncertainty. It depends on what your default position is, what your default decision-making architecture. And I think that's one where we don't really provide the incentives to change the decision-making architecture and then deal with uncertainty in a more quantifiable way.
But Jim, you rather eloquently, I thought, reminded us that we are absolutely and totally certain about the value of preclinical testing when it's positive. And yet we have no real data to support that. Do you want to develop that a bit?
Actually, I didn't say that.
No, I know you didn't.
We've done our best. Obviously, when I say, but nobody knows the answer, I've tried very hard to get the answer. And you can parameterize the uncertainty. For example, if we throw out, based on non-clinical data, a lot of compounds that never make it to the clinic, we're never certain what would have happened. But you can parameterize that and say, well, we know based on information that about 30% of things cause some adverse event in the clinic. So let's apply that to our unknowns, and then we'll parameterize it, between 0 and 100%. You can come up with some estimates. What I can tell you that everything I've done says that the value of negative data is much greater than the value of positive data. And it simply has to do with prevalence. We fail 15% of the time. So your pre-test probability of getting a serious adverse event in the clinic that's going to cause you to stop is 15%. So if you just randomly pick something, 85% of the time, you're going to be okay. Now, I know that's kind of dumbing it down, but you can parameterize the uncertainty. But the value of negative data is actually much better than the value of positive data. But it amazes me that every single conversation I hear equates to we need to improve the positive predictive value of the data. And what you're trying to do is predict a rare event for which the numbers are working absolutely against you.
If I could add to that, the thing I loved about Catherine's talk and Jim's comments just now is because of the way we think about the drug development pipeline, there are these moments where we take very probabilistic things and we binarize them into 0, 1. So, you get out of phase 1 and that's a 0, 1, but that actual phase 1 evidence might be more like a 0.6 or a 0.2. And if we had a system, now this is complicated, but just to think about it, we're allowed to think about ideas without doing them. If you had a system where you had an integrated probabilistic model going through the entire spectrum of development, you could actually accumulate these probabilities using normative probabilistic methods that have been known for, tens, if not hundreds of years. We don't do that, and in a sense, we're doing ourselves a disservice when we pretend like everybody that gets through phase 1 got through phase 1 with the same level of confidence and evidence. It's just not true, and those probabilities could inform your Bayesian evaluation of data that happens in phase 2, and it might lead to different decisions when you accumulate probabilities. We don't have this framework, generally speaking. It would be complicated to do, and there's a certain familiarity with having these decision points that are binary, but in terms of thinking about information processing and probability theory, it's probably not the right thing to do.
The other thing I'd raise is that it seems to me in small drug companies, there's no binary negative outcome. They're never willing to say we should stop, and they'll go on. It's very different for a big company, without putting words in Jim's mouth, who hopefully have other compounds and could redeploy their capital. But for small drug companies, they're prepared to, they have to keep going with the cards they've been dealt, and they will keep going until the money runs out for the 50th time. And so our models, which sound terrific at the IOM, really are not applied to small drug companies. Is that fair, or am I being too reactionary? Nobody's prepared to comment. Sorry.
I think Derek's on the cusp of something.
No, I was actually slightly nervous about being recorded with what I was about to say.
Never bothered me.
No, I think some of it is that the small companies might actually be making very wise decisions, but it's not actually related to product development. It's related to being acquired by a larger company. There's a huge downside to proving that the one therapy you have is sort of lost in talks. And so you just want to not be seen to have made mistakes and not be seen to have got to the end. And then you protect sort of potential investor value. And that's not, We've had a conversation about whether things are done illogically. Some of that might actually be quite logical, and some might even say that it makes sense from a societal value in terms of sort of net production of pharma companies. It just doesn't feel very good for patients.
It speaks to Jim's incentivization, which is right. Just the
Well,
wrong incentives.
I think when you talk about those incentives, you also have to factor sunk cost bias into the equation. So I learned that when we talk to chemists about new safety technologies, if you get them at candidate when they've already invested 5 years, they're really, really skeptical. But when they're pre-lead, they come in and go, I don't want to drill a dry well here. So the incentive in a small company and large company can be very different based on where does the sunk cost bias creep in. You know, I can't possibly have gotten this far and give up versus I'm getting ready to start and I want to make best use of my capital.
Right. Well, one of the big differences is that you don't have to tell anyone about things in the public domain, whereas small companies probably have to declare most of these things immediately. Are there comments from the audience that? Yes.
Hi, I'm Richard Platt. Those were great presentations. I'd like to put also on the table the fact that data is really only good, really good for the purpose that it was originally collected. And when we want to repurpose it for these other uses, it usually takes a great deal of curation. In the experience of Sentinel is that we spend millions of dollars curating data. The problem is more severe for EHR data than it is for claims data. So there's a bright future for data, but I think we always have to be attentive to the fact that it usually takes work to make it fit for some other novel purpose.
Richard, there's lots of literature, or a fair amount of literature now about the copy and paste problems in EHR.
I'm sorry, about
Copy and paste problems in EHR.
Yeah.
If you read patient's record, it's remarkable often how the same spelling mistake is replicated multiple times in the record. I must say that gives me significant pause about thinking that we can discover really important things from the medical record, unless it's something barn door, obvious, like that can't be changed.
Well, the problems are even less subtle than that. I mean, when we started to work with platelet counts, we found that across the Sentinel data partners who had those laboratory test results, there were 67 different units of measure, just for how platelets were recorded. Not the number of the actual value, but just the units of measure. Is it per ml or per cubic millimeter or per nanoliter, and is it in thousands or millions, or is it uppercase or lowercase? And you can say that why is this? It's because even though there are national laboratories, they are built from legacy labs, and there was no particular reason to change them because clinicians can interpret almost every one of those results, so they're useful for delivering clinical care. But if you're trying to do any kind of analytical work across them, you have to harmonize them all, and so it's doable, but it's a lot more work than I think we usually appreciate until we actually start to try to do it.
Hi, I'm Danica Marinec-Dabic, Director of Epidemiology at FDA, CDRH, Center for Devices. Well, I'd like to state a little bit opposing opinion about the repurposing of the data. I actually think that the future is in repurposing of utilization of the data that's already collected. And I think our society cannot afford anything but putting a lot of effort in figuring out the more innovative ways in reutilizing the data that's collected as a part of the clinical practice. So obviously we're going to be spending these 2 days in discussing many of these efforts, but one thing that I would like to put on the table, which I think it's a must, and especially given what Rich just talked about, is that we have to invest in our future scientists, meaning that before we start fixing what's not working and how we can actually use this data that currently exists, we should be thinking in a very pragmatic and practical ways how we penetrate to the schools right now at the medical school, schools of nursing, other schools, PhDs program, and do it very systematically, because that's going to be probably the most effective ways to get your data in a way how you would like to be using it in the future. So that's something that I see is obviously said in conferences, but not everyone is actually implementing this. So I think it's really important step forward from my perspective, making sure that the next generation of scientists is trained now. Right.
Okay.
Brian Alexander from Dana-Farber and Harvard Medical School. I just want to build on a point that Dr. Altman made just recently, which was this loss of information between kind of phases. I think is a really, really important issue, and it may be difficult to kind of parameterize and more formalize information flows, but if we don't do it, it's happening anyways. So, and it really in particular what is happening is biomarker development, where you have some preclinical evidence of a predictive biomarker, and then there's either kind of, we're going to look at all patients biomarker positive and negative in trial, or we're going to do a basket trial or something like that. And there's really nothing in between. So whether it's Bayesian priors or some other kind of information that, you know, if we don't kind of harness the information we have and kind of bring it to bear and see how the information from the trial kind of helps us understand it better, then we're really actually implicitly doing that anyways by making the decisions we make in terms of eligibility criteria for trials.
Okay. Next question.
Hi, I'm Mark Gorman. I'm a cancer survivor and a patient advocate. And I've been both actually for a number of years now. And I've been to lots and lots and lots of meetings at the IOM over the years. So I know you want to talk about regulatory science in the context of how things are handled in development. But I'd like to ask how, kind of in a parallel way, and this does get back to the question of incentives, this change in methodology is actually going to be made so that it can be translated in a way that clinicians and patients can actually trust the new products which are going to be coming onto the market. You know, this is a fraught area, frankly, and right now, just attaching big data to it is not enough. I mean, something I understand pretty well that I think is really being abused is the pitch of the potential for genomic sequencing in a way that it can lead to deterministic choice of outcomes in cancer today. That's just not real. And yet there's lots of incentives that are driving that. So, you know, how can people, society at large, but particularly clinicians and patients, actually come to trust this? And I think there's an implementation science question lurking here. So I'd like over the next day and a half people to kind of give some thought to that and talk about it.
So what do the panel think we need to do before we implement, to pick up on that very important point? What do we need to do to transition to new technologies, and how do we make sure that people are comfortable that they've been adequately validated? Excuse me. Is that a fair summary?
That's a good start.
Okay.
I think these are great, these are salient points, and they do come up when you're sitting in an exam room with results that you need to then turn into decisions. The good news, so I'll do the good news and the bad news. I'll start with the good news. The good news is, maybe I'm too much of an optimist, but I do believe that our ability to collect data will give us pretty good estimates of the probabilities of success and failure for different treatments if science is done well. The bad news is that I do not have confidence that physicians and patients are prepared for the kind of probabilistic reasoning that we might be demanding of them. This this drug has an 80% chance of giving you a 20% recurrence rate or whatever. That is not easy to parse. That might be the form in which the data comes initially, but we really do have to figure out, and this is really, in computer science, we would call it a user interface issue. We have to boil it down. We have to take complex inferential processes, and we have to boil them down to information tidbits that a physician and then a patient in parallel and together can understand. So that is a huge challenge, and it frankly is not a research agenda that is embraced particularly well by anyone. User interface research is very hard to get funded. I'm using user interface as a general for how do you go that last mile with information. It's very hard to get funded from NIH because it's very hard to evaluate. So unfortunately, the way it happens now is it's all market driven. It's who has the product that the doctor or the patient believes is presenting them the information in the most digestible format. And the degree to which that has been appropriately vetted is always unclear. So I think one of the highlight action items would be efforts at making sure that the information delivered to physicians, practitioners, as well as patients is that last half mile or 30 yards needs to be done much better than it is now so that the concepts of probability and utility are captured and translated. So that's not an answer, but it's a research agenda.
But Russ, how has that changed? If you look at the definitive trials that were used to approve a drug, the percentage of patients who responded in these trials is usually fairly small. Physicians, when they use the drug, believe that every one of the patients they treat are going to respond to that drug. We seldom, if ever, I certainly have never said to a patient, I don't think, that there's in the trial, 20% of patients responded to this drug, so we're going to give you that, and we'll see if you're in that 20% or not. Physicians believe it's a graded response when often it's not. So that probabilistic issue that you're talking about seems to me really one that we're now getting better data on, but has existed since time immemorial, and we've just failed to articulate it properly before. Is that fair?
Yes, it was easier to do under a paternalistic model of healthcare, where just trust me, I've come from the ivory tower, and I will be making the best decisions for you here today. We now have a consumer-aware patient base that is not satisfied with the just trust me, or at least they will trust but verify, and then the verification step is what we now need to work on.
Yeah, I think there is work being done in those final 30 yards or whatever, but it's usually not around communicating around drug efficacy. So there's quite a rich research portfolio around communicating end-of-life decisions. It's informed by a lot of social science, a lot of sort of behavioral decision-making, and it's nicely isolated the fact that physicians are probably never going to be very good at it. And it's a huge uphill battle to try to suddenly, quite quickly, train patients to suddenly understand how to understand those estimates, but then it sort of recognizes that there's two humans in the conversation, and it nonetheless works out what can be taught to the physician to be a better facilitator of the decision, given the imperfect nature of the information. The problem is, I haven't seen much of that going on about communicating drug efficacy. So there is some work in the field, but it needs to sort of move over to communicating about drug efficacy.
And the back end of that is communicating risk. So when I looked at your nice diagram of maybe 5 or 6 options, helping a patient understand that the risks associated with each of those pathways are very different, and you're not simply trading the same efficacy for the same risk. And I'm reminded every time I talk to my faculty that what I say, what they hear, and what they understand are very different things.
Jeff?
So I want to amplify on that point. I don't know if I should speak in a Scottish brogue, given the panel composition or not, but...
Keep working on it.
Well, they can understand me in Glasgow. I think that's actually a critical point. And one of the things about this regulatory issue is what's at stake for the patient. What are his or her options? Where are they in their disease? Because people are willing to take much different risks if they're looking for a drug to treat their 7-year-old's runny nose versus their 70-year-old's mother with terminal cancer. And I think that's the thing that I've always found was missing, at least in these broad overviews, is how do we put in this in the equation? How much risk is the patient willing to take as he or she fights an illness, and how do we bring them in the decision? I can tell you from kind of working both ends of it, both the outpatients with illnesses that aren't going to kill patients and inpatients where they are, is that in the latter case, people are willing to take pretty big risks if there's a chance, and in the former case, not. So what do we do to quantitate that in a Bayesian sense? Or do we even need to be Bayesian about it? We can get into an argument. How do we get the regulators to listen to the patient side of it? Do we need to do what the AIDS people did and march in the streets?
I think part of that is the you have to elicit that from the patient population, and I think there are informal ways of doing that in a clinical setting, and then there are more formalized ways of doing that are these kinds of quality of life questionnaires where people with devastating illnesses are asked to make trade-offs. You know, would you rather do this or would you rather do that? And you go through a list of that and you'd ask a gabillion people and then you evaluate those results. And I think between those two things, you start to get at it, but it's definitely patient-driven. It has to come from the patient.
Interestingly, there is a literature now on what physicians choose. And it's counterintuitive to what you think. They're not choosing the most invasive things usually, surprisingly.
Hi, Eric Horvitz. So we all know that we have a continuing big concern with extremely rare but serious adverse effects of drugs that typically will escape the three phases of clinical trials. And we only see the signals in the large reports, large-scale data analyses that come later. With access to large amounts of data and with methods that are being used with success to see signals even when very rare in large populations, might it be time, I just want to get a reflection from the panelists, might it be time for a more explicit, deliberative, designed phase 4 post-approval for studying and reflecting about safety, efficacy of medications when they're actually in the field, where the field is characterized and we actually use statistical methods rather than keeping things sort of in the wild fairs, reports, and staying ad hoc. Is it time to get formal there? Quick question to the panelists.
Eric.
I think so. Where I get caught up on this a little bit is I still think of a phase 4, for example, a phase 4 RCT is testing a hypothesis which is usually framed around benefit. And one of the tricks here is trying to work out, I mean, how do you power for a safety issue that you don't know what it is? I guess you could have some minimal, like some sort of Six Sigma readout for how safe a drug could be. And then you could say that you need to demonstrate that something doesn't happen at some rate. But I feel like safety doesn't lend itself well to RCTs, even though...
No, I didn't say call it RCTs. I said,
so you just mean phase 4, but not
for a new kind of deliberative analysis, given time since release to large populations where we understand the size of populations, we get a sense for priors, and it's less in the wild but more focused and analytical. We have the method. This is one area where big data with the small b and d might have an effect on how we think about post-drug surveillance.
Yeah, I think lots of us
Any
think
other
that
comments
would
on this?
be...
I see the biggest problem as not the rare events that occur, the extraordinary rare events that occur due to drugs, but the common events that occur due to drugs, where the event rate is increased by even a relatively small amount by the drug, and has much more profound public health effects than the rare events. And right now we have no system to detect that, neither in the pre-approval setting or in the post-approval setting, as we discovered to our cost with the non-steroidals and the evolving story around that, around these, I guess. So I see the major problem that we have, not just the rare events, but much more important from a public health point of view, the common events that are increased by even relatively small percentages, which will have profound public health consequences.
Sure, but I guess I was referring to events rare enough that they would more obviously escape the first set of.
Sure, I'm not
Yeah, yeah.
disagreeing. I'm saying that I'm supplementing.
I'll just
Hail
say that I
Marys.
am enthusiastic about those possibilities. I wish and I think that one of the incentives is you could imagine changing the calculus for approval from phase 3 into phase 4. You might be able to make some approvals at phase 3 that you might not make if you had confidence in a phase 4 system that had some teeth. So I think those go together, and it's also in a funny way related to that accumulative probability issue that I was discussing before.
Susan Ward, following up on precisely that, it seems to me that we always position the problem as probabilistic going forwards. So from early discovery to then and to, GLP to clinic to marketplace. And maybe in terms of measuring, which I think I couldn't agree with Jim more about the importance of measuring. It seems to me we should start at the end. And phase 4 would be the best place to start at the end, because that gives you the gold standard, if you like, of what the facts on the ground are. And then back away from that, you know what the answer is, you back away from that and ask what actually tracked to it, as opposed to doing it the other way around, all the way back. And I think that, we might find there's some discomforting questions that we run into when we simply look at the difference between in practice and clinical trials. I'm sure that's not a new thought. One just very specific thought about the question you raised on sharing performance of tox assays. I think that a way to optimize your ability to do that would be to have an independent third party do the analyses with each of the companies. You get away from bias by doing that. And the data would be shared with everybody, but in aggregate, so that no individual company can have a finger pointed at it, but the spectrum of performance can be shared.
And we are going to have some discussions in later sessions on examples of that with CPATH and various biomarkers that have been analyzed from multiple submissions. We're running short of time, so we've got 2 more questions, so if they can be brief.
Kyle Myers, CDRH. I wanted to respond to the point that was made about incorporating patient perspectives and bring you some awareness on initiatives in CDRH that attempt to take this question on seriously and objectively. And the first is to say that we did put out a guidance earlier this year on the incorporation of quantitative assessments from patients of their benefit-risk trade-off, and we have partnered with the Medical Device Innovation Consortium, who at the very same time put out a framework document. We've also stood up for the first time ever a patient engagement advisory board, which will be a board that will allow patients to bring their perspectives to us. This is very unusual. You're probably familiar with our advisory boards that provide us important expert opinions around devices that might be brought to market or general topics. And we involve patients as non-voting members in those kinds of discussions. This will be the first time where we have patients who constitute the advisory board, and industry will be a non-voting member. And as you said, there's a literature on tools for assessing patient preferences. What's really important is that we all get better at communicating quantitatively so that what we say they hear, and that we have the ability to get information back from them. Because you're right, we are always talking about probabilities, and we understand that it's hard for patients to appreciate those probabilities and for us to get accurate, consistent input from them on what their preferences are. Thank you.
I would just add to that on the probability thing, because I'm a risk assessor and I have to deal with probabilities a lot. One area where people really seem to understand probabilities quite well is sort of gambling and sports and those kinds of things. So a lot of times, if you change the context a little bit or something like that, then you can actually communicate more effectively because people really do, they sort of hone in right away on what they understand there. And so if you just draw the parallels, that can sometimes be helpful in terms of probability communication, numeracy.
Yes, Frank Weigold, FDA, work in the Office of the Chief Scientist. Thank you, Karl Myers, for making the point about patient preferences and incorporating the benefit-risk assessments and the scientific aspects thereof, which is very important. And I think next to all the quite important scientific discussion that we have, I think we shall not stop to point out that we cannot accept data and data set fragmentation and inconsistency in data standards, because that is a big problem. If claims data are different from electronic health records, then we can make little progress. And that's certainly not something in the focus of regulatory science, but it is a very important message that we have to make. And ideally, perhaps we should arrive at a point where any person treated for any condition generates data, or data is generated in that context that at least is good enough for something that we would probably call a observational trial or part of a epidemiological study. And as long as we don't do that, we are missing out.
On that note, and I guess Denmark has done something similar to that. On that note, let me thank on your behalf the speakers on the panel, and note that we're going to take a less than 15-minute break and be back at exactly 10:30 to start the next panel. Thank you very much.